Hi, Ms. Blair, and welcome to my cell signaling project on type 2 diabetes mellitus. Type 2 diabetes is a chronic metabolic disorder characterized by the body's inability to effectively use insulin, a hormone produced and secreted by the pancreas that travels through the bloodstream, resembling endocrine signaling. Under normal conditions, insulin binds to specific insulin tyrosine kinase receptors on the surface of target cells, particularly muscle, liver, and fat cells. This binding triggers a complex signal transduction pathway inside the cell, causing autophosphorylation of tyrosine residues. Then, insulin receptor substrate proteins are activated and serve as docking platforms for the PI3K pathway with P55 and P110, leading to production of PIP3, a key signaling molecule and secondary messenger that recruits PDK12, activating AKT, which in turn phosphorylates AS160 and facilitates the fusion of GLUT4 containing vesicles with the plasma membrane, enabling GLUT4 transporters to facilitate the uptake of glucose from the bloodstream into the cell, ensuring proper glucose homeostasis. In type 2 diabetes, cells fail to respond properly to insulin, otherwise known as insulin resistance. The inability of the receptor to recognize insulin can be attributed to mutations in the receptor, obesity, or too much visceral fat around organs. Specifically, IRS proteins fail to get phosphorylated, decreasing PIP3 production and AKT activation, which in turn impairs GLUT4 translocation and hence glucose transport. Normally, AKT inhibits gluconeogenesis, or glucose production in the liver, contributing to elevated blood sugar levels. Due to the inhibition of AKT through insulin resistance, gluconeogenesis may take place in type 2 diabetes. High blood sugar can damage blood vessels, nerves, and other organs in the body. In this scenario, positive feedback can be demonstrated since the response feeds into amplifying the stimulus. Additionally, over time, the pancreatic beta cells responsible for insulin production can become overworked since they must secrete more insulin to compensate for hyperglycemia, leading to impaired beta cell function. Finally, insulin resistance can lead to reduced glucose uptake and metabolic dysfunction, contributing to neurodegeneration as the brain relies heavily on glucose for energy. Under normal circumstances, IDE degrades insulin and the amyloid beta monomer. In diabetes, the levels of circulating insulin are higher, and hence most of the enzyme is utilized to degrade insulin, allowing amyloid beta to aggregate into plaques. These plaques are the hallmark of Alzheimer's disease, the most common neurodegenerative disease worldwide.